Hi, and welcome to the Science in the News lecture series, Fall Lecture Series 2015. We are going to get started in just a couple minutes with our second lecture of this 9-part series, The Internal War on Cancer, Using the Immune System to Fight Tumors. During the lecture, please feel free to use the form below to ask questions or to give us feedback on your viewing experience. Again, we'll get started in just a couple minutes. Thank you for joining us.
Getting started today, we wanted to make sure people were filing in due to all the traffic in the rain, and thanks to you for braving the rain and being here today. My name is Vinny, and this is...
And I'm Kelsey, and we are the co-directors of Science in the News. Science in the News is a graduate student organization here at Harvard University, and our main focus is to really bring non-scientists and scientists together to have productive and educational lectures and events on various science topics. So we've been having our fall lecture series now for 15 years. We're celebrating our 15th birthday, and we're really excited about the lineup this year. We have a great schedule for you. This is our second lecture of this series. And if you haven't gotten one already, I would encourage you to pick up one of these flyers on your way out. And it has the full schedule on the back, but basically we will be here every Wednesday at 7 p.m. with a different topic. Before I forget, I want to thank our sponsors who make all of these events possible for us and also provide the yummy snacks. So we're sponsored by the Harvard Coop, BGSO, the Graduate Student Council of the Graduate School, and Harvard Medical School. And I want to tell you guys a little bit more about other events that we do. So in addition to our lecture series, which Vinny will talk more about, we also have a spring version of our lecture series, and that occurs on the Cambridge campus, and we'll be starting in about March, so be on the lookout for that. In addition, we have a really fun event called Science by the Pint, which I would also encourage you to grab a schedule. These are monthly events occurring the second Monday of every month. So the next one will be coming up on October 13th. And what we do is we invite a scientist and their lab members to a bar, specifically the Burren in Davis Square, every month. And the scientist gives a short talk about what they do, and then they spend the rest of the time with their colleagues, mingling from table to table, talking very casually about science, answering any questions you might have. So it's a really fun event. I'd encourage you to check it out. We have Dr. Nikhil Nayar with Engineering New Microbial Life Forms on October 13th. In addition, we also have an online newsletter we call Signal to Noise, where we publish articles on various science topics, and these are released 4 times a month. So now Vinny will talk about what tonight will be like.
All right, so I see a lot of familiar faces in the audience, but for those newcomers, I'm just going to go over the format of tonight's lecture and what you guys can expect. So generally during a Science in the News lecture, we have the lecture split up into 3 separate parts, which go about 20 to 30 minutes each. And within each section, we pause for questions a couple of times with a specific question slide. So we ask that you hold your questions until then, and they'll be pretty frequent as well. And if you notice the 3 lecturers repeating your question, it's because we have a lot of viewers, hopefully on the live stream. So hello to those people. This is so they can hear your question as well and still be a part of this. And they can actually ask questions on their own as well through our online form. So these 3 young gentlemen today are all in immunology labs, and they'll be talking to you today about cancer immunotherapy. So they'll first start with an overview of the immune system, and then talk about the immune system and cancer, and then finally go into some of the newer therapeutics. The other thing I want to bring to your attention is we have these handouts with some terms and some useful links and recommended reading on the back. In addition, we have these surveys, which we really like for feedback and for any further topics or future topics that you guys would all like to see in lecture series in the coming years. So with that, we will turn it over to Yerne. Enjoy.
All right. All right. Thank you, Vinny. Can everyone hear me? Is that all right? Great. So you heard a little bit in the introduction what we're going to talk about, which is the immune system, and particularly how the immune system interacts with cancer, and how scientists and clinicians now are utilizing some of this new knowledge to actually treat some of the cancers using the immunotherapy. Before we begin, does anyone know who this person is? Good, I wouldn't have known either if I didn't look him up last week. Turns out this is Dr. William Coley. He was a prominent surgeon in New York in late 1800s. And what he was doing was something very interesting, quite bizarre actually. He was actually treating his cancer patients using live and dead bacteria. What he saw is that if he actually injected some of these bacteria into the tumor itself, he saw the tumors actually shrinking and sometimes disappearing altogether. At the time, Dr. Coley was ridiculed for the science. The scientific community really didn't buy that there's anything real to it. But throughout the last 100 of years or more, what we know is that Dr. Coley was really doing the first kind of approaches to cancer immunotherapy. What he was doing is he was actually boosting the immune system by giving it that foreign adjuvant, something that the immune system has not seen before, and it recognized it as foreign and got a cue to attack that region of the body. And as mentioned, over 100 years later, cancer immunotherapy is considered one of these latest and greatest breakthroughs that is applied in the clinic today. And Azan actually next part will go over this in more detail. So Vinny mentioned a little bit, but just to go over it, of the series of events today for this talk, we separated in 3 parts. My name is Yarne, and I'm gonna start off by introducing what cancer is, what the immune system is, and how these 2 systems interact together. Azana will take us through a little bit of the history because it has some very interesting history of why we know now that the immune system is actually very important in cancer. And then Vikram will give us a brief overview of the latest immunotherapies that are actually being applied in the clinic now. So what is cancer? I think we all have heard of cancer, probably know someone that has or is struggling with cancer. But in terms of the scientific lingo, we think of cancer as uncontrolled expansion of abnormal cells. And what does that mean? Imagine you have a healthy cluster of cells in your body. It can be anywhere, and through everyday exposure to various stressors that can be from the environment, some of these cells are starting to accumulate some mutations in their genome. And what is a mutation? It's really a permanent and random change in the DNA, so in the genetic material of the cells. And this DNA really encodes the blueprint for all the cell machinery, so all the proteins that are this machinery. If we imagine it that a protein or particular functional protein is a key, a mutation can cause the key to change shape slightly. So this key is now not opening the right door anymore and can actually start opening the wrong doors that will have some detrimental effects. It's important to know that these mutations happen all the time, and actually most of the mutations are not harmful at all. They just cause the cell to die right away, or they actually cause a silent mutation, so it actually not change the key at all, and the function remains the same. However, certain mutations, particularly in the cell machinery responsible for cell division, cell survival, and cell death, when that machinery is mutated and abnormal, some of these cells can actually become cancerous. So this is just showing that one cell turns cancerous, starts forming this tumor by proliferating, expanding dramatically without any control. And what can happen if the cell mutates even further is that the cell becomes so different from its original tissue that it actually leaves the tissue, spreads throughout the body, throughout the whole system, and that's what we call that the cancer has metastasized. They have spread throughout the body. So that's a quick blurb about cancer. Now let's talk about the more interesting part of the biology. Granted, I'm slightly biased being an immunologist, which is the immune system. And immune system is a very complex system that has many different cell types playing a very specific role, and each of these cell types has to work together with every other part of the system to be optimal and actually functional. So I just listed a few things, a few different cell types here. There are actually many more. For example, we have the natural killer cell, or the NK cell, that can directly kill some of the abnormal cells. We have the B cell that some of you may have heard already because they produce these protein calls called antibodies. And these antibodies are very important with protecting us against certain viral infections, for example, by directly binding the virus and preventing the virus entry into the cells. These antibodies can also be engineered now to bind to specific molecules, and some of the therapies are now used to manipulate that pathway. Then we have the dendritic cell that is constantly scouting the environment, scouting the different tissues, and looking for any dangers. Once it sees a danger, it needs to alert the rest of the immune system that something is going wrong, that the other branches of the immune system should get activated, recruited, and clear the particular abnormality. We have the macrophages and neutrophils, which are really the big cells that eat up cell debris, for example, from dead cells. They eat bacteria, so really clearing up the tissue and restoring normality. And we have the T cells, and the T cells are really the prime cell type that we use in cancer immunotherapy at this moment. There are many flavors of T cells. One T cell type in particular that is very important is what we call the cytotoxic T cell, which can directly kill abnormal, so infected or cancerous cells. And as I'll go through in some next slides, the T cells can't really do much without the help from the dendritic cell. All right, so the immune system, we really think it has evolved to protect us from foreign invaders. So these foreign invaders can be the viruses, bacteria, fungi, any kind of infection, anything that does not actually belong in a healthy tissue, in a healthy body. So you can imagine we can have a virus that is going to enter our system. Let's say it's a respiratory virus. It's going to enter through the nose, infect some of the epithelial cells, so cells laying the respiratory tract. The virus enters the respiratory tract, infects some of these cells, and actually hijacks these cells, making the cells practically be a factory of virus that produces viruses. And eventually, the cell is going to burst, spread all these viral particles that can then infect the other cells. So this is really bad. We don't want this virus to be in the system. We want to get rid of it as soon as possible before it spreads throughout a lot of the tissue. So I mentioned before that we have the dendritic cells then that are continuously scouting the environment, looking for any particular danger. And the dendritic cell is actually very quick to see that there is something foreign in the system. So a virus does not belong, has never seen it, and it gets alarmed very quickly, and we call it gets activated. So the dendritic cell knows that it needs to, it can't just clear up the virus itself. It actually needs the help of the rest of the immune system, particularly the T cells, for which the dendritic cell actually needs to travel to the lymph node. So in the lymph node, the naive T cells reside. So the T cells are naive because they haven't been activated yet. So they're waiting around, looking for the right infection. It's important to know that each of these T cells, and I'll go over it a little bit next slide, but each of these T cells is actually recognizing a different part of a different virus, for example. And it needs to see the right signal for the right T cell to actually be activated. And this is a very hard process for the T cells to actually find the right protein, or what we call antigen, that we're going to go over. But when it sees the signal that recruits the T cell gets activated. And by activated, the T cell starts multiplying to make multiple copies of itself, to make a whole army of these cells. It also starts making various effector function molecules. For example, cytokines, which are the soluble mediators that will come up in the next part of the talk as well as some of these the molecules that can directly kill abnormal cells. So once multiplied, these T cells need to travel back into the site of inflammation, in this case, in the site of the viral infection, recognize quickly the cells are actually infected with this virus, and destroy these cells. So the cells can mostly regrow quite easily, but we want to basically burn down all the factories that are making this virus. So the T cells, by killing all these infected cells, are restoring balance in the system again. I mentioned that this activation of the right T cell is actually not a trivial step. And that's because part of it is that you have so many different flavors of naive T cells, where basically each of the T cells you have in the body has its eyes set on a different part of a protein. So this can be a viral particle, a bacterial particle, anything. And the way this happens is this T cell progenitor cells, it's a type of a stem cell. In the process of developing the T cell pool, this progenitor cell actually undergoes genetic rearrangement in a specific part of the genome that is responsible for this specific T cell receptor to be made. So in the end, you have a pool of T cells, each one recognizing one particular thing. So I mentioned then it's from the millions and millions of cells that you have in the body, it's actually very hard to find the right dendritic cell that is giving the right signal to get activated. So for this, the T cells are actually traveling from the lymph node through the blood to different lymph nodes again, looking for a particular red flag in this case that will activate it. And the dendritic cell is doing the same. It's scouting the tissue to look for dangers, but then once it sees a danger, it needs to go back to the lymph node to find the right T cell. And that helps creating this army of activated T cells that are ready to attack. I mentioned also that there are other parts of the activation steps that regulate this process. For example, so this is just a magnified interaction of the dendritic cell here with the T cell. To actually get the proper activation, the T cell first needs to see the right, what we call antigen. So in this case, a part of a virus through its T cell receptor, TCR. In this case, it has the eyes for the right color of the flag. So this gives it the first signal that you need to get activated. This is not enough, however. The T cell also needs to see this other interaction of CD28 on the T cell with B7 on the dendritic cell. And well, we won't go too much into details here because it can get very complicated very quickly. It's important. This is an important regulatory branch that oftentimes prevents us from autoimmunity, for example. So the dendritic cell can be presenting your own healthy cells flags, but you don't want to activate those T cells because those T cells can actually start attacking your healthy cells in that case. So this molecule B7 only gets turned on when there's a real danger associated with the signal. The immune system, to prevent partly the autoimmunity and also to get rid of all these T cells once the viral infection is cleared, has developed negative stimulatory pathways as well. So two in particular we'll focus on today are through this molecule called PD-1 and the other one called CTLA-4. And the real reason why I'm bringing these two up right now is that they're very important in cancer immunology as well. So tumors have actually evolved to use some of these molecules to inhibit these T cells themselves. So that they think that it's okay, don't kill me, and they can survive much better. So at this point, I basically just went over the whole immune system in 3 slides. So I would imagine that there are some questions out there. So I'd love to take any questions if so. Anything? Nope. Oh, I'm sorry. Yep. Correct.
And then once you figure out what you want, does it also create the
Yes, so this progenitor is what we call, it's a thymocyte because the T cells are developed in the thymus, and the thymocytes, there's a handful, not handful, couple of thousand, but it's definitely fewer than the actual T cells. But as these T cells, or thymocytes, go through the different developmental stages, they undergo this rearrangement of the T cell receptor to make it different. And it is basically every thymocyte can self-renew as well to create more and more of the different T cells. Does that kind of make sense?
Yeah, I guess 2 years ago, all the 10 billion one cell really between
It's not quite just one, but it's a very small number of these cells that give rise to all of the T cells down the road, right? Correct. Yeah. Sorry, yeah, I realize I need to repeat the question. So how does the T cell actually develop if it has never seen a virus or bacteria? Is that a question? So this rearrangement in the genomic region that is responsible for the T cell receptor is actually completely random. It's making random T cell receptors, hoping that one or a few will be the right ones to actually recognize the virus, and at that point, it will get those particular T cells that just happen to have the right receptor that is binding to the viral particle. That T cell is going to expand and make the effector cells.
From the live stream from Atlanta. What is the method for T cell deactivation once the infection is cleared, if there should be?
Yes. So the question is, what is the method of deactivation after the infection is cleared? So this really comes down to some of these inhibitory pathways, particularly CTLA-4 is very appreciated in down modulating the immune responses. So the T cells undergo this massive proliferation or cell division after the activation, but then you're right, you need to get rid of all those cells because there's only so many T cells you can have in your body. So you need to clear those cells once they're no longer needed and give room for other T cells fighting other infections. So viral infections in general don't really fail to be noticed by the immune system. Different viruses have evolved. Sorry, so the question is, why do some viral infections, if they don't get noticed by the immune system? And the answer is really that they do get noticed and by the immune system and boost this response, but some of the viruses have evolved other ways to inhibit this immune response to the virus using various different things. So sometimes you see this red flag that the dendritic cell is putting up that is showing that that it has seen the virus. So the infected cell would have the same red flag up. So the virus can actually kill that red flag. So the cell is incognito in there because it's no longer presenting any flags on its surface. So, but that's usually the second layer of the immune recognition.
The symbol of potential T cells is primarily generated in childhood. I mean, kind of completely active in adults, right? I mean, how much toxicity do you maintain as adults?
True, the thymus shrinks a fair bit, almost all the way in a healthy adult. However, there is some growing literature that there is still some remnants of the thymus that is left and can give rise to some of the T cells. But most of, at some point it is believed that the heterogeneity of the complexity of the T cell gets lower and lower because you don't have new input of new T cells.
Which means that the system does not learn.
So the system does learn, actually. That's something that I didn't really go into, but once the T cell gets activated and expands a lot to kill all the virus-infected cells, a small fraction of cells doesn't get cleared, and those are the cells that turn into memory cells. And those cells can actually persist, in some cases, for the rest of your life. And that's, for example, how some of the vaccines work. They give you a mock infection to activate the T cells. The cells expand, but then a fraction of them turn into memory cells, and those memory cells can then be activated very quickly when you see the real infection. So you never really develop the symptoms because the infection gets cleared right away.
But then what you think that for learning, it is necessary for learning that the deactivation is effective?
No, it's.
Some of those are created are not destroyed, so
Yeah, so this was a simplified model of how this works. In reality, not all of the activated cells, not all of the cells in the army are created equal. We believe that some of them may have from the very beginning a predisposition to become memory cells. So they may have received slightly less of a signal, for example, or they have been in the right environment to actually make memory cells down the road. But this pathway is actually the same in all of these cells that you want to clear them with. Did that answer the question? All right. Any other questions? Okay. Say it again. In
Like for organisms that do not have an immune system as developed as humans, is the frequency of seeing cancer less or
so organisms are not mammals, or
like all organisms, anything that is evolved?
so every healthy organism should have immune. Sorry. So the question is, do organisms without an immune system would it have more cancer? And actually, Azana will go through this because it has actually been a bit of a contentious topic in the field through history with genetically engineered mice that have or don't have certain branches of the immune system. I'll let him answer that part. But in general, every healthy organism that would persist in the wild would have an immune system because it's really just so hard to survive otherwise because we're exposed to microbes on an everyday basis that you know it's like a story of the bubble boy that you have to be in this enclosed bubble because the microbes are all around us. So in a human situation, it would be really hard to address that. But Azana will talk about a mouse situation. All right. Good. So I told you that the immune system has probably evolved to protect us from foreign invaders, but in a very similar way, the immune system actually can protect us from cancer as well. And the way this works is that you have again your healthy cells, and eventually some of these cells will turn cancerous, and you'll have this tumor growth. The dendritic cell is going to go around and scout the environment again, looking for danger. However, in this case, it's actually very hard for the dendritic cell to see the danger because the cancer cells actually your own cells that have slightly changed. There's certain proteins that are now a little bit different, but there's nothing really, really foreign in this case. So the dendritic cell is confused, but we believe that in most cases, it does actually get activated. It gets alerted and raises up the flag, putting up the particular fragments of the cancer cells, again, travels to the lymph node to find the right T cell that will get activated upon seeing that particular fragment of the cancer cell. Again, the T cell will expand, make a whole army of effector cells, and these T cells will travel back to the site of the tumor to eradicate the tumor, to literally kill off the tumor cells. However, even though I tell you that this is how we think that the normal system works, we all know that cancer is one of the biggest health burdens in the world. We all know someone with cancer. So the system is clearly not perfect. It's far from perfect, which is why we have such a cancer prevalence. And that's really because the cancer has evolved many different ways to actually evade the immune system. So there are more than three, but I'll just present three different ways of how this can happen. For example, the first way is that the tumor cells are really too similar to your healthy cells. So again, we have the dendritic cells scouting the environment, looking for any danger, but it never quite gets beyond this stage of just being confused that there may be something going wrong, but I don't think there's anything worth alerting the rest of the immune system about. And this is actually very important because if it would get activated about a healthy cell, then you would have autoimmunity. So autoimmunity would be where your T cells would be activated to start killing off your particular cell types. For example, in the case of type 1 diabetes, when the T cells are abnormally educated to start killing some of these insulin-producing cells in the pancreas. So a different way is where the dendritic cell has actually gotten activated, has recruited the T cell. The T cell has gotten activated, made a whole army of soldiers that travel to the site of the tumor. But the cancer cells have adopted some of these ways that are actually inhibiting these foot soldiers. So, for example, one of the ways is by expressing this molecule PDL1 that I mentioned before that can directly send an inhibitory signal into the T cell to deactivate it. The third one is, in some ways, one of the more interesting ones, I think, is where the dendritic cell has seen the virus as foreign, has activated the immune system. The T cells have actually came into the tumor and actually cleared most of the tumor. However, a small fraction of the tumor cells have mutated slightly further. So they're no longer expressing this particular protein that the T cells are recognizing. So, for example, before, let's say they were recognizing some red protein, but these tumor cells are not red anymore, they're purple now. So the T cells actually don't recognize, and they don't really know what to do with them because they're specific for the red tumor. And after this, the dendritic cell can come in again, can activate a second cohort of foot soldiers that would travel in, but the tumor can mutate more again. So you have this constant coevolution of the tumor always being one step ahead of the immune system. And this lets it grow out as well. So I'm going to stop here. I'll take any questions, and Azana will go on after me to talk a little bit about this historic view of what a contentious topic it has been through the history and why we know that all this is actually very important. So any questions? Please.
On the previous slide, the tumor cell to someone is healthy cell. Does that mean that people who have autoimmune diseases are less likely?
So the question is, are people with autoimmune diseases less likely to get cancer? I'm actually not sure about that. Horizontal, would you know about that? Just come very close.
Yeah, so it's a very good question. And so the basic answer is that we're not sure, but there are kind of two competing things. So when you have autoimmunity, you have, for example, T cells that are attacking. He mentioned in diabetes T cells that are attacking the pancreatic cells, and so in that case, you'll have a lower incidence of tumors in the pancreas. But because the immune system is kind of focused on this autoimmunity, then it's kind of distracted from other things. And so you will kind of see this incidence of other tumors. So there's that balance that it's not entirely clear which how often this happens. That's actively being studied, but that's kind of the prevailing thought that you won't get tumors in that particular tissue, but you're more likely to get them elsewhere. Yeah, Azan will talk more about the idea of having a chronic inflammation, which means that you have an ongoing immune response, and how that can also stimulate tumors, or that can also lead to tumors. And so that's a case where perhaps in the long term, they are more likely to get tumors at that particular spot. So it becomes quite complex. It's not so much that if you have autoimmunity, you're preserved from getting tumors.
Of those recent answers you explained, could you explain a little bit better an insight of what would that be the case? Because since you were invoking like adaptation, so it seems like at some point then you would come to the conclusion that cancer is benefiting the organism to reproduce more and more, right? Because still the cancer cells I would assume have the same DNA and the rest of the cells. So if they evolve to do that, because there's some adaptive benefit, so
There's adaptive benefit. Sorry. So the question is, since the cancer cells are expanding more, if that is an adaptive thing for a beneficial thing for the organism, is that right? So it's really beneficial only for the tumor itself. It is an abnormal thing that can actually outcompete the whole system for resources, for real estate in your body, and that can actually cause death in these patients. But it is actually a constant adaptation, and the reason why, for example, even in this case of inhibiting the immune system, why most of those cancer cells would survive is because all the other ones may have gotten killed, right? So, but it is a benefit for the tumor cells themselves to get eventually selected to only have those that have adapted to evade the immune system. Right. All right.
Over here.
There's a lot of hardware.
Yeah. It's like going to space station.
This one goes into the pocket.
Goes in the pocket. Okay. One sec. There we go. Okay. Hey everyone. It really is a pleasure to be with you today. Yarne gave a really solid introduction. It's a very hard task to fit 2-3 years of classes into 30-minute presentation, so solid job. What I'm going to talk about has more to do with the history than the actual science, so it's more of a history lesson. So I want to start with this lovely guy Yarne talked about. And he was a doctor, and his patient was a teenage friend of John D. Rockefeller Jr. So I couldn't find a picture of her, and we can assume she looked something like this. She was in the same social circles. So her story has somewhat of a sad ending. She had a swelling in her arm. Initially, they thought it was a... Oh, is this working well? Can you hear me? Initially, they thought it was an infection. But when they biopsied her arm, they noticed that she had a particularly aggressive sarcoma, which is a type of cancer that comes up from your connective tissues in your arm. So they tried to amputate her arm, but she still passed away because her cancer had metastasized. And Coley, he was a 28-year-old fresh-faced surgeon, and he was really affected by her death, and he was inspired to look for new treatments for cancer. So while digging around, he gets an idea. He hears about a patient who had an aggressive sarcoma as well, like another tumor. And this patient gets another bacterial infection while still in the hospital. So you can imagine back then, it's not as clean. A lot of people are getting infections willy-nilly. And the patient gets this infection, and he survives the infection, and his tumor magically disappears. Coley actually really wanted to make sure, and goes and finds the patient in the streets, sees for himself that the tumor has regressed, and starts thinking, maybe bacteria actually fight tumors and kill it. So there's no clinical trials or anything. He finds patients, he injects them with bacteria. It works sometimes. It doesn't work some other times. So there was a lot. It was very hot in the field. The people saying, "No, you're wrong. You're killing these patients with bacteria," and people arguing that you are right. This is working in some patients. And some doctors saying, "Oh, we see the bacteria eating up the tumors." That's probably wrong. We know today that cancer, the reason it worked was because of some sort of non-specific immune activation, as Yarne mentioned. You basically throw a grenade in there, and you turn the immune system on, and the immune system clears the tumor. And because of this, he's known as the father of cancer immunotherapy, even though he had no idea what he was doing. He had no idea what he's doing had to do with anything to do with the immune system. But he did say something really profound. He said, when people were arguing with him, he said, oh, the day will yet come when posterity will be amazed that we remain so ignorant of things that will seem to them so plain. And yes, he is right, actually. Now, if you go to any immunologist, you go to any classroom, you tell the students, oh, did you know, read all about it, the immune system fights cancers. They'll say, yes, we take it for granted, we know. But the question I want to answer today is, how did we get there? It wasn't written in somewhere, in some text like in the Bible or anything. It took a lot of work to get there, but how did we get there? And to answer that question, I want to introduce this concept, the concept of a beam balance. So in science, whenever there are two competing ideas, what happens is both sets of ideas are judged based on the experimental evidence that they provide. So that's how ideas become dogmas. That's how it becomes paradigm. If you believe in something, you need to show the experimental proof for it. And you put proof for one idea on this side and one idea on this side. And then it's weighed out and people decide, okay, this is the correct idea. So the first idea is the immune system has a protective role in cancer, right? So basically what that means is, as earlier mentioned, this lovely T cells will see a developing tumor, come back, swords unleashed, and fight it off. The second idea is that the immune system has no protective role in cancer. Basically, your tumor goes crazy, does what it wants. The immune system, T cells, dendritic cells, they have no idea what's going on. They have no role. So two hypotheses. And people started to look for evidence supporting either one. Does the immune system play a role or does it not? So the first piece of evidence supporting the hypothesis the immune system has no role came from observations in the clinic. So as when you were answering the question of this gentleman, we alluded to, there are some types of immune responses when they're chronic, they sort of correlate with higher incidence of cancer. So why this happens is really quite complex, but I can give you clinical examples. So let's talk about HCV infection. So what happens is your liver, you get infected through sharing needles or something. The virus gets in your system, it gets into your liver, infects the liver. Unfortunately, HCV is not cleared. It sticks around with your liver for a long time. So you have a chronic immune response to it. This progresses to cirrhosis or formation of scar tissue and ends up as liver cancer in 20, 25 years. So you have a chronic immune response and then you develop cancer. And also there are similar observations with autoimmunity. For example, if you have Crohn's disease, you have chronic inflammation in your intestines, in your colon, you're really predisposed to developing colon cancer. So these are observations that people made, and they said, okay, the immune system has no protective role in cancer. Let's rule that out. Second piece of evidence supporting this hypothesis comes from a humble mouse. So this mouse is known as a nude mouse. It's obviously the name nude means it has no hair. What basically happened was there was a farmer. He saw this mouse in his farm, naturally freaked out that it might damage his crops and brought it to the laboratory. We immunologists are not interested in it because it has no hair. The reason we're interested in it is because this mutation has a spontaneous mutation which results in it having no T cells, completely no T cells. So over here, just to make sure that nobody's falling asleep, I want to ask you a question. What do you think is happening in this mouse when it comes to tumors, to cancer rates? Is it higher or lower, or no change? So who says higher? You can raise your hands. Who says lower? No change. So sort of a trick question. It is no change. There is no change in the cancer rate. It's the same thing as a regular mouse. So now, if I were to end my presentation at this point, you'd say, what they were talking about? The RNA was giving us all this evidence, or like telling us T cells are important for cancer, but then there's a mouse with no T cells and he has no change in cancer rates. The immune system has no role. So, and that's exactly what people did. They said, oh, look at this mouse, no T cells, no change in cancer rates. Case closed. Let's move on. Immune system is now where we're supposed to look when it comes to looking at cancer biology. So, but at this point, the balance had shifted in this direction, supporting this second hypothesis. There are tumor-promoting kinds of inflammation or immune responses, and there is no increase in nude mouse cancer rates. So, two strong pieces of evidence. But then, as time went on, during the late 20th and early 21st century, other works started to come out. First observation, you remember this nude mouse, right? Didn't have any T cells, but it still had a cohort of all the other immune cells Yarny mentioned. And cancer immunosurveillance is such an important evolutionary concept that nature has made sure there are several redundant pathways protecting us, or this mouse, from cancer. So it really, the absence of T cells, although is bad, does not predispose this mouse to increase cancer rates. So to test this question in mice, a group in St. Louis genetically engineered new mice. So basically, you modify the genome of the mouse in ways that we can discuss if you're interested how it happens. But they genetically modified mice so that they are severely immunodeficient, more immunodeficient than the nude mouse, or they lack more cell types. So over here, you have a normal mouse, screen, solid immune system. And as it ages, just like human beings, some mice do get some sort of intermediate cancer, but a good 80% of them, they're cancer-free at this age, which shouldn't be surprising. They have all their immune cells. But if you take away two of the cell types, and you look at mice similar age, none of them are cancer-free. And almost 40 to 50% of them get advanced cancer. And then if you look at another mouse, where you take away not just two, but 4 immune cell types, the majority of the mice not only just get intermediate cancer, they get advanced cancer, and there are no mice that are tumor-free. So, what this really nailed, what this experiment showed definitively is, in the mouse, the immune system plays a role in protecting us from cancer. So that's all nice, that's perfect for the mouse. But as you know, human beings are not just big mice. There's fundamental differences. What is the evidence that the immune system plays a role in cancer in human beings? And that evidence comes from another simple experiment. What a group in Pennsylvania did is they looked at patients with ovarian cancer. And I know the ovaries are not up here. That's just the model we have. But so they looked at a group of patients with ovarian cancer. And they noticed that some patients have T cells in their tumors and others don't. So they thought, okay, what happens if we just follow these patients? And it's exactly what they did. They treated them the same, and they followed their patients. And to explain the results, I want to introduce to you the concept of a survival curve. So the survival curve basically is on the vertical y-axis, you have the percentage of patients that are still alive. And the x-axis, you have the time that they're still alive. So what you can clearly see is the patients that have no intratumoral T cells, these guys, by 5 years, unfortunately, they all pass. But the patients that do have intratumoral T cells, they survived long after these guys have all passed away. So it's key. The key point here is there is no difference in these two patients except the T cells. So in humans, really, you can't get any clearer than this. This is very convincing for a human study. If you're familiar with literature with human studies, you know it's really hard to see an effect in humans because there are so many differences. They're so diverse. And it's not just genetic mouse. You inbreed them, you keep them, you treat them the same. But human beings, there's so many differences. So it's hard to see a strong effect like this. But this was a really strong effect and had the field really, really excited. Okay, so to conclude the first section of my talk, I want to summarize what I just told you all about. I told you about all the evidence that caused the balance to shift this way and make sure that the idea, the hypothesis that the immune system has a protective role in cancer is the accepted dogma today. The first piece of evidence is that the nude mouse is not completely immunodeficient. And then the second piece of evidence is that severely immunodeficient mice that were genetically engineered, which actually is a lot of work, really, to make those mice get significantly higher rates of cancer. And the third final piece of evidence being that patients with intratumoral T cells survive much longer and have a much better prognosis. In fact, there's a lot of debate in the field whether we should use the presence of such cells in the tumor as a prognostic marker to tell us how well the patients are going to do and predict what their outcome is going to be. Yeah. So I would like to open up the floor for questions at this point. Question?
Going back a couple of slides to the survival curve.
Yeah.
I guess the question is, why in the first place some people have intratumoral T cells and others don't?
Yeah.
Is that you can tell me why they survive in the first place?
Yeah, that's why they survive. Sorry, the question is, repeat the question. Your question is, why do these patients get this intratumor T cells? Or is that why they survive better?
Why do some patients have intratumoral T cells and others don't? Why in the first place?
Yeah. Yep. So repeat the question is, why do some patients have intratumoral T cells and why do other patients don't in the first place? So that is a really great question. Short answer is we don't know. And we're working on it. And Yarday and Vikram in their labs, it's an active art of study. Why are these differences? And even if you look at some of the stuff that Vikram will present with clinical breakthroughs in clinic, some patients respond to particular treatment, some patients don't. Why that happens is an active area of study. Why some patients have intratumoral T cells and why some patients don't is also an active area of study, and we don't know. Any more questions? Sure. The earlier slide? Yes. So these observations were made decades ago. Nude mouse observation was made in the 70s, like 74, I think. This one's actually, speaking of science in the news, this is really all really recent. This one was in 2001. These two was one discovery. And this one, I think, was in 2003. So it's only within the last few years that actually people really started to believe that the immune system has a role. In fact, Vikram was relating to me a story of how the guy who made this discovery came to this very room to tell the story, and he was, really strongly questioned about it 2 years ago. Question?
The question from the live stream is
Yes, that's a good question. Forget to repeat your question. The question over here is routine testing of intratumoral T cells being done? Yes, that is being done. And actually for some tumors, intratumoral, the presence of such cells is used as a predictive marker for how that person is going to do in the clinic. Rather than looking at how invasive is the tumor, you just look at are there such cells in the tumor, and if those cells are there, then you can predict a favorable outcome for the patient. It is being done. So actually, we did that last week in our lab. So,
yeah, but I would follow this comment. How would you say that the patient has more T cells inside the tumor with metastasis, while the patient doesn't have that, or have metastasis with less T cells, it would be better?
Yeah, we're doing is what we basically do is you take a biopsy, and then you look for the presence of such cells in there. Obviously, sorry, let me repeat your question. Your question is, are there other confounding variables? Can you just, instead of just, it's not just as simple as there are T cells, there are no T cells. I agree. It's not a perfect science, and it's not 100% predictive as well. So there are other confounding variables. It's not just, yes, cells are there, you're perfect, you're golden. And even the patients with T cells, only 20-25% of them survive. So it's not 100% predictive. There are other confounding variables. I do agree with that. Okay. Question? Yes. So the naked mole rat, it's actually there was a really nice study done on it. It wasn't comparing it to a nude mouse, but they did a really nice study on it, and it doesn't have any cancer. Well, cancer rate is much lower, and the reason it does that is because it expresses some sort of receptor on itself, on the surface of its cells, and that inhibit out of control proliferation. It's called CD44. And that was a recent study that came out, I think, a year ago, and it might be something interesting there. But yeah, that's why that mouse doesn't get any cancer. It actually has very little to do with the immune system. It's nothing inbred in the mouse. So, right. Okay.
So is it possible that the methods of altering the immune system components in the mice also induce other unknown changes that affect their responses?
Okay, that is a really great question. And it is highly possible that the methods you use to genetically modify the mice, but also highly unlikely because of the extensive work that has been done in this mice, trying to look for other things that are responsible. But I can't say with 100% that all the work that you did didn't change something else. But so far, there's nothing that's come up. Right? Does anybody have a better answer? Move on. Okay, so part 2. So at this point, I would like to sort of conclude the history lesson and tell you more science. The first section I want to talk about is how are tumors identified as being different from your own body cells. And the second part I want to talk about is how are excessive immune responses kept in check. So 2 parts. 2 actually very related parts, but 2 parts nonetheless. Okay. So to answer the first question, what does the immune system see? I want to describe to you an experiment that was done a while ago, like 20, 30 years ago. What they did was they got red tumors and they weakened them. Basically, you slap on the handcuffs. Either you irradiate them or you mutate them. Some way you weaken your tumors. And then we went to our favorite mouse model, favorite laboratory animal, the mouse, and you gave it these weakened tumors to this mouse. Weakened red tumors. So you wait a little while, and then you challenge this mice, split them into two groups. The first group you challenge with red tumors, and the second group you challenge with green tumors. So what will happen to these tumors? I think we've done the whole drill. We can move on. We don't need to. But what happens here is your red tumor is rejected, but your green tumor actually grows. So this experiment tells us several things. You can make several conclusions after looking at this experiment. The first conclusion you can make is that there is a specific response against the tumor, which means you gave them red tumors, you made a response against the red tumor, and there's only a specific response against this red tumor, not against this green tumor. Red and green, they're totally different tumors. First observation. The second observation is the immune system can distinguish between a normal cell and a cancerous cell. As you're having a response against the red tumor in this mouse and rejecting it, but the mouse is not dying, its normal cells are fine. How does this happen? How does your immune system tell this apart, tell these three, your normal cells from the red tumor and the red tumor from the green tumor? Well, this happens, if you remember Yerne's earlier slide on how cancer is generated or how cancer is induced by an accumulation of mutations. You have several mutations, which means you're changing the blueprint, genetic blueprint of yourself, and as a result of changing the genetic blueprint, you're changing what they produce on the other end, or the proteins that they make. So these proteins that they make that are changed, that are different, those are what we know as tumor antigens. So basically a tumor antigen is the end result of the change in the genetic makeup of your cells. Because you got a mutation, you made a new protein, and you made a tumor antigen. Your immune system is so well trained to recognize foreign material, it actually can tell the difference. It's just a small, subtle difference between a cancer cell and a regular cell. Small, a change in one or two proteins, you can pick it up. So, to summarize what I just told you, this genomic instability or mutations result in the production of new proteins. Also, in addition, it results in the overproduction of some proteins. So if your immune system is used to a certain threshold, if you go past that threshold by overproducing certain antigens, your immune system responds to that. Or also, this genomic instability or mutations change in the blueprint of your cells in your body, results in the expression of proteins that are where they're not supposed to be. For example, if there are certain cells that are exposed in a developing embryo, certain proteins that are expressed in developing embryo, and they end up being expressed in an adult body, your immune system has never seen them in an adult, it responds to them. So these are the things that allow your immune system to tell the difference in the red and green tumor and your normal body cells, also known as tumor antigens. But then, once your immune system can see these tumor antigens, how do we make sure that you have the perfect response? And that's done, to sort of elaborate a little bit more on Yerne's slides, is done through the use of different checkpoints. So you get the perfect response. You have a go sign in your immune system, in your immune response, which is CD28, that's the technical term for it. You have the stop sign, or CTLA4, and the slow sign, PD-1. So these three things combine to give you an immune response that's just right. Not too hot, not too cold. It's like Goldilocks porridge. But then what happens if you get rid of this? This is a key concept for Vikram's section as well. But what happens if you get rid of the different checkpoints? What happens if you get rid of your stop sign? In mice, so genetically engineered mice, what happens is 3 weeks after they're born, these mice die from excessive response against their own bodies. Because there is no stop sign, their immune system goes crazy. They get severe autoimmunity, which is basically a response against self. And this is a survival curve. Regular mice are born, but a permanent loss of the stop sign or CTLA4, and your mice are dead in 3 weeks. They're all gone. And this was actually work done in your name Vikram's Mental's lab, like 2 years ago here at Harvard. But then, what's this pathway, the relevance of this pathway for cancer is that a temporary loss of the stop sign, so to describe this experiment to you, what they did was they injected tumors into mice, and then they temporarily took away the stop sign in this group of mice, the black bar, and then for 3 days, and then looked at, okay, what happens to the tumor? There is a clear difference. The tumor in mice and normal mice, in which the stop sign is kept intact, that tumor grows excessively. However, in mice where the stop sign is temporarily removed, the tumor is much better controlled and doesn't actually, a lot of them clear it completely. So the key point of it here being a temporary loss of the stop sign or CTLA4 can protect you from cancer or can protect you from, can clear tumors, while a permanent loss is actually fatal. But then what happens when you, and then the second part is the slow sign. You have the stop light, the stop sign that brings everything to a grinding halt, but then you have the slow sign that sort of maintains it, maintains everything, make sure the immune response is right, but it's not as dramatic. If you look at the mouse model in which this slow sign has been removed, you actually see something interesting. As I mentioned earlier, the mice that are deficient in the stop sign, unfortunately, they die from an autoimmune response within 3 weeks. But the mice in which the slow sign is removed, they're fine, they're healthy. And actually, when they first made this mice, they started thinking, oh, well, this mouse is still alive, it's fine. Maybe this slow sign or PD-1 is not that important. But its importance is actually shown by an experiment that someone did in Atlanta at Emory. The experiment they did was they infected this mouse with a virus, with a chronic virus infection. And what they noticed is, although a normal mouse survives its infection, a mouse that doesn't have this slow sign, once the immune response starts to this virus, there is no slow sign, it overspeeds, goes crazy, and the mouse is dead in a week. So this experiment nailed the importance of the slow sign. The stop sign, I showed you, the mice are dead in 3 weeks after being born. But this one right here, PD-1, the importance is underscored by this experiment where you need to give them an infection, you need to kickstart the immune response to show its importance. So a running theme of this talk is that tumors have ways to take advantage of this stop and slow signs and use that to evade the immune response. And this is really an important point for you to understand what cancer immunotherapy is. And Vikram will explain that more in detail. Okay, so as a conclusion, I want to end by telling you, basically summarizing what I told you in the last 30 minutes. So I told you in sort of a roundabout way, by giving you a small history lesson, that the immune system plays a role in cancer. And then in a more direct way, I told you that the immune system has tumors that express antigens, that tumors have antigens that the immune system can use to recognize that they are not normal cells. I also told you that the immune system uses several checkpoints to ensure that you have an appropriate response to every challenge. So when a patient comes to clinic, the immune system has obviously failed it. And Vikram tells you how we can shift the balance, which means how we can make sure that we shift the balance towards the immune system away from tumors, and how you can clinically intervene to do that. Okay, last slide, I promise. I wanted to underline this one fact to you. So this stop sign or slow sign PD-1, it took 20 years from the identification of the gene for us, over 20 years, for us to appreciate that, okay, we can use it in cancer and for the FDA to approve it. From 1993 to 2014, from 1987 to 2011. And it all started with basic research, curiosity-driven research, where people were saying, what is this gene? What does it do? And this really underscores the importance of such work, and also tells us not to be short-sighted of the things that we support. And as PSA, make sure your representatives are also not short-sighted as well. Right. Thank you.
So we're actually running a little low on time. So we'll take one question and then we'll take like a five-minute break. And then you can feel free to come and ask the presenters more questions at that time before we go to the third part.
Okay, questions? So your dendritic cell, it picks up the antigen. It has no idea what it is. It takes it to a T cell, and it's your T cell that says, oh, this is something foreign. And just like you have a T cell specific for the virus, you have a T cell specific for this tumor antigen. That's what happens. Okay, let's take a break. Like, please feel free to come down, ask me questions, ask any of his questions. Did I go over? Did I go over?
I apologize that it was a bit short. If you have any questions, we'll be around after to answer them. And also, I think we'll be on the lab tour, so if you have any questions there, we can answer those as well. So in the first part of this talk, you learned about how the immune system responds to cancer. In this part of the talk, I want to talk about what happens when that response fails. And I want to talk about some really exciting therapies that have come to the clinic in the last decade or so that utilize the knowledge that Jörn Anazan had told you about. Before I do, I just want to quickly recap the basic points from the first part of the talk to really drill them home. The main point of the talk that I hope you've taken away is that for the most part, the immune system responds to cancer. And how does this happen? Well, when dendritic cells, which are constantly looking for foreign things in the body, see a tumor, this looks kind of foreign, and so they get activated, and then they find the T cell that has eyes for the tumor. They activate this T cell, which means that they make it take up arms, grow out in size and number, and then send it on its way into the tumor, with the hopes that the T cell will then be able to clear the tumor, restore order. But of course, as Azana mentioned at the end of his talk, when a patient comes to the clinic, this implies that this response has effectively failed. In some way, the T cell was unable to kill the tumor, or the other immune cells were unable to recognize the tumor. And so what do we do in this case? This is the basic idea that drives all of the different areas of immunotherapy, that the immune system has failed, and we want to shift the balance back in the favor of the immune cell. And there are two general approaches to do this. The first one, immune cells out of a patient's body, manipulate them in some way, and then put them back in with the hopes that they'll go to the tumor and clear the tumor. The other is, instead of taking cells out, injecting something else in that will manipulate these cells while they're still inside the patient. And so I'll kind of go through a cartoon of how each of these would work. So in the first approach, as I mentioned, the idea is to take cells out of the body. So for example, you can take a T cell out of the blood and take it into a petri dish where scientists can manipulate it in some way. In this case, they'll manipulate it so that they take up arms, multiply a number, and hopefully become very potent so that when you put them back into the patient, they can go to the tumor and clear it. The other approach is, as I mentioned, to manipulate cells while they're still inside manipulate any cells while they're still inside the patient. And one of the main ways to do this is through the use of antibodies. Jörn brought up this word, and maybe you've heard it before. An antibody is a molecule that can very specifically recognize another molecule, and it can have a variety of functions. In this case, for the most part, when we talk about antibodies and therapy, we're talking about antibodies that can block some particular interaction. In this cartoon, the idea is that the antibodies would be injected in the patient. It would go into the tumor and bind either T cells or tumor cells and block some type of interaction that's happening between them. And I'll go into details on what interactions those might be. But the end goal is that the T cells will stop being suppressed by whatever's suppressing them, multiply out, take up arms again, and attack the tumor. So those are kind of the two paradigms for cancer immunotherapy. They all kind of fall into these two categories. And I'm going to go in depth in one therapy within each of these approaches. So first, I want to talk to you about taking immune cells out of the body, manipulating and putting them back in. But before I go into the science behind this, I want to tell you a story that will hopefully motivate and excite you about these therapies. And this is about a girl named Emily Whitehead. Emily, unfortunately, was diagnosed with acute lymphoid leukemia, which is a cancer of the blood. In particular, her cancer was actually a cancer of B cells, which you've learned about. They weren't acting as the immune cells that Jörn told you about. They were actually just growing out of control. And so at the time, Emily was given the standard of care, which is a chemotherapy. I'm sure you've all heard of chemotherapy. The goal is basically to give a drug that will poison cells that are rapidly dividing. And it's obviously here that includes the tumor cells, but it also includes a lot of other cells. And so I'm sure you're all aware of the toxic effects of chemotherapy as well. In Emily's case, that certainly happened, but fortunately, it also cleared the tumor cells and she was declared in remission. As you might have guessed, within a short period of time here, 17 months, they detected the cancer cells in her body again. And at that time, she was given what's called reinduction chemotherapy, which basically just means that she was given chemotherapy again in the hopes of clearing the tumor. And unfortunately, in her case, that happened. She was declared in remission. They couldn't detect any of the cancer cells in her blood. But unfortunately, this time very shortly after, within 4 months, the cells were detected in her body again. And so at this time, then they attempted this reinduction chemotherapy, but unfortunately, the cells were not responsive. You learned about ways that that cancer cells can evolve to avoid the immune response. Similarly, they can evolve to avoid being susceptible to different drugs. And so her cells had evolved to not be responsive to chemotherapy anymore, and they tried some really strong ones with no effect. And so her parents were obviously quite desperate to find something that could help her. And fortunately, they took her to Children's Hospital of Philadelphia, where doctors there tried this, at that time, and still experimental treatment, where they, as I mentioned, were able to take cells out of her body, manipulate them in some way, and I'll get into more detail about what they did in her particular case. But basically, they engineered them so that they could attack tumor cells and put them back in. And happily, she was declared in remission after just 1 month. I shouldn't say just 1 month, and I put an asterisk here to remind myself of that. This was by no means an easy month for Emily. There are certainly side effects associated with this therapy, and I'll tell you more about those. Her parents may actually be watching this talk, so I want to make sure I don't shortchange that month. But I'm also happy to report that she's cancer-free today, and this is a photo from her website, emilywhitehead.com, and I highly encourage you to go read. Her parents have a blog there talking about this experience and her experience since. So before I get into the more specific treatment that she got, I'm going to tell you about a similar treatment that I think is a little bit more intuitive, and then we'll go into hers. So this whole idea of taking cells out of the body and putting them back in kind of started with the observation that there are already T cells within a tumor, as your name is on, I've told you. But the implication with the T cells being there is that though they may recognize the tumor, they're not able to clear the tumor. And so the idea was, hey, if we have access to these T cells, can we take them out, put them in a petri dish? Obviously, this is not always the case, but if we have access to them, for example, in the case of a metastasis where there's one on the skin, but there's not but we don't have access to all the metastatic sites, maybe out of the one we do have access to, we can take out the T cells, take them into a dish, give them a variety of molecules that will stimulate them to become active again, and take up arms, multiply, and then inject them back in with the hopes that this time they'll be able to go into the tumor in greater number and with greater potency and clear the tumor and restore order. But of course, I should say that this is a very exciting avenue of research and this is being actively studied. But one question you might have is if they failed the first time, what's to say they won't fail the second time, and will they even be able to get into the tumor? And so this is certainly a hurdle that people studying this approach have run into. But out of this idea of taking cells out and putting them back in came this very interesting idea. If we're taking the cells out of the body already, why not manipulate them with other approaches that are being developed for other areas of science? Why not manipulate them in other ways before we put them back in? We already have access to them. Why not just do that? So there's this idea of chimeric antigen receptors. You learned from Yurnay about the T cell receptor, which is basically this molecule that T cells have on their surface so they can go around and try to interact with dendritic cells that have their antigen. They use this T cell receptor to look for that antigen. Well, what if we give these T cells, a receptor that we know is on tumor cells? What if we give them a receptor that we know recognizes things that are on tumor cells? So we basically leave the dendritic cell out of the equation. Instead of having to have the dendritic cell present something and then the tumor cell has to show that antigen as well, what if we just have the T cell be able to directly recognize the tumor cell? And so this is a chimeric antigen receptor. So the idea here is instead of taking T cells out of the tumor, we're able to just take them out of the blood. We have access to many, many more of them, and then you don't have the issue of having access to the actual tumor. So what if we take out this T cell, and then while we have it in the dish, we can do the same thing as before. We can activate it, have it take up arms, multiply out, but now we can carry on the further step of basically giving it eyes for the tumor. As I mentioned, we can put a receptor on its surface so that it recognizes the tumor. And so the idea then is that once you have enough of these, you can inject them back into the patient. And the hope is that they'll then traffic to the tumor and clear that tumor. So this is, I think this is a very elegant and interesting idea, and I like it a lot because it kind of brings together many areas of science. But one issue that maybe you have in the back of your mind right now is, how is this that different from the normal immune response? You still have all these other barriers. So for example, you have these suppressive mechanisms that tumor cells can evolve to prevent the immune response. You still have that. You're just putting in cells that you've manipulated in some way. And so I think this is really the exciting part of these CAR T cells, these chimeric antigen receptor T cells, is people that were working with these had the idea, hey, if we're already manipulating them in some way, why not just take it one step further and try to interfere with this interaction? So the idea is actually quite simple. It's again, you take cells out of the blood. Again, you give them this cocktail to make them multiply and take up arms. Again, you give them specificity so that they attack the tumor. But this time, you mess with this normal pathway that they have. So normally, they see the stop sign and they think stop. So this is what's happening inside the tumor. Well, what if while we're giving them these receptors, we also give them the ability to, when they see stop, they think go. And so actually then when the tumor says stop, the T cells become stronger. So the idea, of course, then is you inject these in, these now more potent cells get into the tumor, and the tumor thinks that it's really smart and it's clearly going to say stop, you're not going to be able to attack me. The T cell is now actually stronger and is able to clear the tumor. So this is the approach that, that Emily got, and this is being actively studied. In fact, there are already 14 published trials using this CAR T cell approach. Those 14 trials encompass almost 100 patients. So it's obviously a relatively small number, but we can start to draw some conclusions from these. And there are more than 20 ongoing using different permutations, using different types of approaches with subtle differences between all of them. Primarily, they're looking at blood cancers. I kind of showed you this whole cartoon as a solid tumor. I think that's just a little bit easier to intuitively grasp, but the idea is the same in the blood, that you're still taking these cells that are very specific for then now the blood tumor cells. The idea is analogous. And one of the really exciting observations is that many of these patients have their blood cells, the blood cancer cells, cleared within a month. And I should note that different tumors, different cancer cell types do respond differently. Emily, as I mentioned, had ALL, acute lymphoid leukemia, whereas in patients that have chronic lymphoid leukemia, CLL, they're less likely to respond to a therapy like this. And so why that is currently being studied. It's too early to make a call on long-term effects. There are some patients that did clear and then relapsed. Happily, there are many, many responses. But as I mentioned, this is all in the last 5 to 10 years, primarily in the last 5 years. And so it's too early to make a call on the long-term effects, but it's very promising at this point. And as I mentioned, I would come back to Emily's story. So during that month, what ended up happening is something you don't see in other types of cancer trials. That's kind of an emerging theme. I'll come to that also in the second part, is that there are effects with these immunotherapies that we do with normal cancer therapies. And so one of the other areas of investigation is how to deal with these other side effects that clinicians are just not used to dealing with. So the one here, the predominant one that was seen in these CAR T cell trials is this thing called cytokine release syndrome. Yerné introduced the idea of cytokines. Basically, they're these molecules that immune cells release out into their environment in the hopes of stimulating other immune cells. So one of the side effects that they saw was when they injected in these CAR T cells, they had such a potent response that that so many cytokines were released either by themselves or by other immune cells that were already in the patient, that they stimulated, that they set off other immune cells to act. And there was, for example, flu-like symptoms, and then Emily started to have some respiratory issues and then some cardiac issues. Fortunately, her doctors recognized what was going on and were able to give her neutralizing antibodies. And so through these trials, for example, with Emily, doctors have kind of realized what kind of side effects to expect and have been able to deal with them. But this is a very interesting one that you won't see primarily with other cancers. So before I move on to the other general approach to immunotherapy, I'll pause here. I'm sure people have some questions. I'm happy to answer them or try. Okay, so the question was, when I talked about you turning off this, taking away the stop mechanism, or in fact turning it into a go mechanism, doesn't that have other effects? And that's a very important observation. And I'll say two things. One is keep that question in mind, because it'll definitely come up in the second part of the talk. But the other thing is that keep in mind that, well, you can't see the eyes here, but keep in mind that these T cells have eyes specifically for the tumor cell. And because of this specificity, hopefully they're not going to be attacking other cells. It's certainly a concern. And so, not to get into too much detail, but one of the things that people are now putting into these T cells is what's called a suicide mechanism, where you can give some type of molecule so these cells all kill themselves. These T cells that you put in, the idea is if you do start to get a crazy immune response against yourself, you can just give this molecule and these cells will all die to prevent exactly what you're talking about. But for the most part, we think that the specificity that you have from the T cell receptor will prevent that from happening.
We have a question on the live stream. So does each tumor have a specific antigen associated to it, or is it different all the time? And if so, why is there a recurrence of cancer and if an immune response has previously been activated for that same tumor? Is it a mistake with the immune system, or does that only happen if the tumor is?
Should I repeat that? Okay, yeah. Okay, I won't repeat the question, but I'll try to answer it and summarize. So the basic idea is that, as Yerne and Zana have mentioned to you, there are these tumor antigens. It's certainly not just one. So when a tumor cell becomes a tumor cell, it becomes very mutated. If it just had one mutation, it's very unlikely that it would be that bad and that would grow out of control. So by the time we actually see a tumor in the clinic, it has a whole lot of mutations that have led to it growing out in this crazy way. And so along with having these things that allow it to grow out, it also has mutated proteins that it presents as antigens. So it certainly has many antigens. And so you'd hope that you also have many immune cells that respond to those antigens. And if the tumor, this is an interesting question, because if the tumor was completely cleared, could you get a recurrence of exactly that tumor cell? I would hypothesize no, you wouldn't. But what likely happens is that you don't actually get a complete clearance of the tumor. There are still some residual cells. And as Yerne mentioned, the red tumor can be almost completely cleared, but there are a few cells that turn purple, so they change these antigens so that they're not recognized by the immune system, and those ones can grow out. That's one thing. Or the tumor cells can figure out, hey, if I start to use these mechanisms to suppress the immune response, now I can grow out. There's this constant battle, as we keep on alluding to, there's this constant battle between T cells and tumor cells. And so even if we don't see the tumor, you can have, even if we don't see it on the macroscopic level, even if you don't see a bump, or if you don't see it on a scan of the body, there can still be small numbers of tumor cells still there. And those are the ones that we think grow out again, that are now able to avoid the immune response.
Hi, sorry, I might have missed it earlier. Is it one T cell per antigen? Is it one? Yeah, is it one T cell? Does one T cell one antigen?
Yeah, so it's kind of, yeah. So each T cell recognizes very specifically one antigen, and for the most part, only that antigen. And there's pretty much one there's a single T cell that'll circulate around the body looking for that antigen. Once it sees it, it'll grow up and it'll form many. Good question. So the idea, let me actually jump back to this idea of the adoptive transfer. So in that case, you're taking one or several, whatever number of cells you can get out of the tumor, and growing those up with the thought that those ones have eyes for the tumor and those ones will attack the tumor. So you're limited to only what you can get and whatever you can actually grow out. In the context of this chimeric antigen receptors, they will have eyes for something that they're specific for, but you actually override that. You don't care what they normally see. I'm portraying this as one cell growing out, but you'll actually take out thousands or millions of T cells that all see very different things. But since you're engineering them, you don't really care what they see. You're saying, I'm going to override that. I'm going to give them different receptors so they have eyes for the tumor. And then now I can do these other manipulations. So that's, I think, the really beautiful thing about it. You're not really limited to what the tumor's to what the T cell's normally seeing. You can override that whole system. Okay, so the question was, are you creating a memory response when you make these chimeric antigen receptor T cells? Irene told you before that when T cells respond to viruses, they grow up in large numbers so they can clear off the virus, and then once the virus is gone, then they dampen down and only a few cells remain. Those are memory cells. And the answer is yes, you're making a memory response in this case. What I just said was that you take out thousands or millions of cells out of the blood, kind of with no thought for what those T cells already are or do. Then you may be taking cells that are already memory cells that already attack the virus, that have already attacked the virus and are memory cells for that virus. You may be taking naive cells. And so that's one of the issues that you run into is you don't necessarily know what the T cell has seen in the past. That's actually being actively studied. Would it be better to start with something that's a memory cell? Would it be better to start with something that's a naive cell? And you can kind of debate which, but the real answer will come from the experiments that are being done in labs, and then presumably move on to clinical trials. Okay. One more. So the question is, you engineer the chimeric antigen receptor, so there's something called VR. Yeah, so what that is basically one of the regions of the T cell. Your name mentioned that you have these receptors on T cells that randomly combine. So basically you just get the random receptor on itself, and one of the highly random parts is what you're talking about, VR, three regions basically. And the answer, no, you're not changing what you're doing. You're saying you can have whatever cell receptor already had. You can keep that. But I'm going to add a new one on there. I'm kind of making and so, as Anna mentioned to you, this idea of CTLA-4 blockade. Again, CTLA-4 is one of these stop signs that's on T cells, and when it's engaged, it thinks, okay, I got to stop. And he also presented to you some data in mice to show that if you block CTLA-4, then tumors grow more slowly, or in some cases are cleared. And now I want to present to you some clinical data that just came out in 2010. And so this is still one of the most exciting figures from the field of cancer immunotherapy. It's really the one that's driven the last several years of research. Again, this is a survival curve. So on the y-axis, you see the percentage of patients that are still alive after some period of time that you see on the x-axis. And in this case, they took patients that had advanced metastatic melanoma that stopped responding to everything that was given to them. And so, as you'll notice here in the control arm, they had a very poor prognosis. Within only a year, 75% of patients had died, and within 2 years, you were down to 10% still alive. So obviously, this was a very sick patient cohort that had really no other options. And so in this clinical trial, they either gave them the control treatment, the standard of care at the time, a molecule that will block CTLA-4, so take away the stop sign, or a combination to see if there's some benefit to having both of them. And so what you'll see is, as I mentioned, this line, and this line, the patients had very poor prognosis. Here, this may seem subtle, but you have a shift of both of the curves in the upward direction. And what this upward direction implies is something actually very profound, and it drives a lot of the interest in this field. And this is something very unique to immunotherapy. What you see is that you have a relatively few fraction of patients that responded in this particular case. So you went up from about 10 to 20 or 25% of patients still alive. But this is very unique, this flat line right here. What this implies is that though there were very few patients that responded, they responded very durably. This goes out to 4 years. As you can see, the curve stops in the control. Before that, these patients still survived. And in fact, some of these patients are still alive today. And so I just want to place this kind of in the context. I'm going to jump out of immunotherapy just briefly into this idea of targeted therapy, just to give you a context for how impressive this curve is. So in this, this is another study that was published around the same time, and the idea was to use what's called targeted therapy. Jorné introduced to you this idea of mutations that happen in the cancer cell, and for example, he presented to you this idea of a blueprint in the DNA that encodes for this key, and if the key gets changed in some way, maybe now it opens up doors you don't want it to open. And in fact, this can cause what are called driver mutations, which means that these are the things that really drive the tumor cell to be a tumor cell. And so the idea was this targeted therapy was blocking this key from functioning with the goal of preventing the cancer cells from being able to grow out. And what you see is, and this is more common in cancer therapy, is that you have many patients that respond. You see after 3 months in the control arm, again, this is very aggressive melanoma, there's only 20% of the patients have died after only 3 months, but most of the patients receiving the targeted therapy are still alive. But unfortunately, after only 9 months, the curves collapse on each other. So what this implies is that many of the patients respond, but it's a short-term response. And so that's why this response is quite profound. This targeted therapy is more representative of what you normally see. There are a few other lessons that were learned from this clinical trial that I think are really interesting. The first is when they injected these antibodies into the patients, they noted that the tumors first grew. And in fact, they were going to cut off the clinical trial there. They thought this is causing the tumor to grow out of control. We should stop this. But after following them just a little bit longer, they saw that the tumors actually shrank. And so there are two quite important points to take away from this. The first one is I said shrank. I didn't say that the tumors went away. In most cases, the tumors were smaller than initially, which is great because they didn't grow. These are patients that, as I said, had very aggressive cancer, so you expect them to grow out of control, but the tumors didn't go away completely. You may have heard this idea of turning cancer into a chronic condition. This is kind of along those lines where these patients may still have a tumor burden, but they're still alive. The cancer is being held in check in some way. And the other important point is that they wanted to figure out why these tumors grew at first. So when they took these biopsies, in the cases where they could get biopsies, for example, on the skin, they looked what happened, what the tumor looked like before treatment and after treatment. And what they noted was before treatment, there were some T cells responding, but there were very few. They were massively outnumbered. But after treatment, they were actually the tumors were dominated by an infiltrate of T cells in the tumor. And so the thought is that actually this increase in size was just basically because so many T cells came in to attack the tumor that the tumors first grew out and then shrank. So that's something that, again, you don't see in other cancer therapies. That's something that's quite unique here. And so those are 2 points that now are kind of being factored into clinical trials. Normally in clinical trials, you think about the tumor size as the endpoint, but here, if you thought about tumor size as being something that you're kind of determining if the drug is working or not, well, then this doesn't necessarily look great. First of all, the tumor's growing at first. That's obviously, that sounds bad. And two, the tumor's not being completely eradicated, which is obviously the end goal, but maybe it shouldn't be necessarily. Okay, so before we conclude, I want to stress a few points. So one is that we've presented to you a small number of signals that T cells see and use to control their activation. So the first one was that Jorné really walked you through this idea that dendritic cells use this T cell receptor, use this interaction with the T cell receptor to stimulate the T cells. So that's kind of the first signal that they see. But importantly, there are other signals that they see. For example, they see these go signs. So when they see both the signal, the first signal, the T cell receptor signal, and also this go sign, then they think, okay, hey, now I should really act. But in order to control this, I walked you through this idea of checkpoints. But I want to overwhelm you with how many other signals there are that are important. Don't try to read these. I just want to stress that there are many of these signals that are currently being studied, both positive and negative. And so these are all different nodes that you can imagine targeting either alone or in combination to try to drive a strong immune response. So these are being studied all around the world. Out of this, I want to really highlight one of them that you've already heard about, and that's PD-1. I'm going to go back to this Goldilocks idea. As Donna mentioned, if you have this very strong go signal, maybe you'll get too hot of a response. When you have the CTLA-4 stop signal, maybe you'll get too cold of a response.